Chapter 5. The Engine of Evolution. What but the wolf's tooth whittled so fine the fleet limbs of the antelope? What but fear winged the birds, and hunger jeweled with such eyes the great goshawk's head? Robinson Jeffers, The Bloody Sire. One of the marvels of evolution is the Asian giant hornet, a predatory wasp especially common in Japan. It's hard to imagine a more frightening insect. The world's largest hornet, it's as long as your thumb, with a 2-inch body bedecked with menacing orange and black stripes. It's armed with fearsome jaws to clasp and kill its insect prey, and a 1/4-inch stinger that proves lethal to several dozen Asians a year. And with a 3-inch wingspan, it can fly 25 miles per hour, far faster than you can run, and can cover 60 miles in a single day. This hornet is not only ferocious but voracious. Its young larval grubs are fat, insatiable eating machines who insistently rap their heads against the hive to signal their hunger for meat. To satisfy their relentless demands for food, adult hornets raid the nests of social bees and wasps. One of the hornet's prime victims is the introduced European honeybee. The raid on a honeybee nest involves a merciless mass slaughter that has few parallels in nature. It starts when a lone hornet scout finds a nest. With its abdomen, the scout marks the nest for doom, placing a drop of pheromone near the entrance of the bee colony. Alerted by this mark, the scout's nestmates descend on the spot, a group of 20 or 30 hornets arrayed against a colony of up to 30,000 honeybees. But it's no contest. Wading into the hive with jaws slashing, the hornets decapitate the bees one by one, with each hornet making bee heads roll at a rate of 40 per minute. The battle is over in a few hours. Every bee is dead and body parts litter the hive. Then the hornets stalk their larder. Over the next week, they systematically ravage the nest, eating honey and carrying the helpless bee grubs back to their own nests, where they are promptly deposited into the gaping mouths of the hornet's own ravenous offspring. This is nature red in tooth and claw, as the poet Tennyson described. The hornets are fearsome hunting machines, and the introduced bees are defenseless. But there are bees that can fight off the giant hornet, honeybees that are native to Japan. And their defense is stunning, another marvel of adaptive behavior. When the hornet scout first arrives at their hive, the honeybees near the entrance rush into the hive, calling nestmates to arms while luring the hornet inside. In the meantime, hundreds of worker bees assemble inside the entrance. Once the hornet is inside, it is mobbed and covered by a tight ball of bees. Vibrating their abdomens, the bees quickly raise the temperature inside the ball to about 117 degrees Fahrenheit. Bees can survive this temperature, but the hornet cannot. In 20 minutes, the hornet scout is cooked to death, and usually the nest is saved. I can't think of another case, save the Spanish Inquisition, in which animals kill their enemies by roasting them. There are several evolutionary lessons in this twisted tale. The most obvious is that the hornet is marvelously adapted to kill. It looks as though it was designed for mass slaughter. Moreover, many traits work together to make the wasp a killing machine. They include body form, large size, stinger, deadly jaws, big wings, chemicals, marking pheromones and deadly venom in the sting, and behavior, rapid flight, coordinated attacks on bee nests, and the larval "I am hungry" behavior that prompts the hornet attacks. And then there is the defense of the native honeybees, the coordinated swarming and subsequent roasting of their enemy, certainly an evolved response to repeated attacks by hornets. Remember, this behavior is genetically encoded in a brain smaller than a pencil point. On the other hand, the recently introduced European honeybees are virtually defenseless against the hornet. This is exactly what we would expect, for those bees evolved in an area lacking giant predatory hornets. and therefore natural selection did not build a defense. We can predict, though, that if the hornets are sufficiently strong predators, the European bees will either die out unless they are reintroduced, or will find their own evolutionary response to the hornets, and not necessarily the same one as the native bees. Some adaptations entail even more sinister tactics. One of them involves a roundworm that parasitizes a species of Central American ant. When infected, an ant undergoes a radical change in both behavior and appearance. First, its normally black abdomen turns a bright red. The ant then becomes sluggish and raises its abdomen straight up in the air like a taunting red flag. The thin junction between the abdomen and the thorax becomes flimsy and weakened, and an infected ant no longer produces alarm pheromones when attacked, so it can't alert its nestmates. All of these changes are caused by the genes of the parasitic worm as an ingenious ploy to reproduce themselves. The worm alters the appearance and behavior of the ant, which advertises itself to birds as a scrumptious berry, and in so doing brings on its own death. The berry-like red abdomen of the ant is raised up for all birds to see, and easily plucked because of the ant's sluggishness and the weakened junction between the abdomen and the rest of the body. And birds gobble up these abdomens, which are full of worm eggs. The birds then pass the eggs in their droppings, which ants scavenge and take back to their nests to feed the larvae. The worm eggs hatch within the ant larva and grow. When the ant larva becomes a pupa, the worms migrate to the ant's abdomen and mate, producing more eggs, and so the cycle begins again. It is staggering adaptations like this, the many ways that parasites control their carriers just to pass on the parasite's genes, that gets an evolutionist's juices flowing. Natural selection, acting on a simple worm, has caused it to commandeer its host and change the host's appearance, behavior, and structure, turning it into a tempting mock fruit. The list of adaptations like this is endless. There are adaptations in which animals look like plants, camouflaging themselves among the vegetation to hide from enemies. Some katydids, for example, look almost exactly like leaves, complete with leaf-like patterns and even rotten spots resembling the holes in leaves. The mimicry is so precise that you'd have trouble spotting the insects in a small cage full of vegetation, much less in the wild. And we have the converse, plants that look like animals. Some species of orchids have flowers that superficially resemble bees and wasps, complete with fake eye spots and petals shaped like wings. The resemblance is good enough to fool many short-sighted male insects, who alight on the flower and try to mate with it. While this is happening, the pollen sacs of the orchid attach to the insect's head. When the frustrated insect departs without consummating his passion, he unwittingly carries the pollen to the next orchid, fertilizing it during the next fruitless pseudo-copulation. Natural selection has molded the orchid into a bogus insect, because genes that attract pollinators in this way are more likely to be passed on to the next generation. Some orchids further seduce their pollinators by producing chemicals that smell like the sex pheromones of bees. Finding food, like finding a mate, can involve complex adaptations. The pileated woodpecker, a crested bird that is the largest woodpecker in North America, makes its living by hammering holes into trees and plucking insects like ants and beetles from the wood. Besides its superb ability to detect prey beneath the bark, probably by hearing or feeling their movements, we're not sure, the woodpecker has a whole group of traits that help it hunt and hammer. Perhaps the most remarkable is its ridiculously long tongue. The base of the tongue attaches to the jawbone, and then the tongue runs up through one nostril, completely over and around the back of the head, and finally re-enters the beak from below. Most of the time the tongue is retracted, but it can be extended deep into a tree to probe for ants and beetles. It is pointed and covered with sticky saliva to help extract those tasty insects from holes. Pileated woodpeckers also use their bills to excavate large nest cavities and to drum on trees, attracting mates and defending their territories. The woodpecker is a biological jackhammer. This poses a problem. How can a delicate creature drill through hard wood without hurting itself? Think of the force it takes to drive a nail into a plank. The punishment that a pileated woodpecker's skull takes is astounding. The bird can strike up to 15 blows per second when it's drumming for communication, each blow generating a force equivalent to banging your head into a wall at 16 miles per hour. This is a speed that can crumple your car. There is a real danger of the woodpecker injuring its brain or even having its eyes pop out of its skull under the extreme force. To prevent brain damage, the woodpecker's skull is specially shaped and reinforced with extra bone. The beak rests on a cushion of cartilage, and the muscles around the beak contract an instant before each impact to divert the force of the blow away from the brain and into the reinforced base of the skull. During each strike, the bird's eyelids close to keep its eyes from popping out. There is also a fan of delicate feathers covering the nostrils so that the bird doesn't inhale sawdust or wood chips when hammering. It uses a set of very stiff tail feathers to prop itself against the tree, and has an X-shaped four-toed foot, 2 forward, 2 back, to securely grip the trunk. Everywhere we look in nature, we see animals that seem beautifully designed to fit their environment. Whether that environment be the physical circumstances of life, like temperature and humidity, or the other organisms, competitors, predators, and prey, that every species must deal with. It is no surprise that early naturalists believed that animals were the product of celestial design, created by God to do their jobs. Darwin dispelled this notion in The Origin. In a single chapter, he completely replaced centuries of certainty about divine design with the notion of a mindless, materialistic process, natural selection, that could accomplish the same result. It is hard to overestimate the effect that this insight had, not only on biology, but on people's worldview. Many have not yet recovered from the shock, and the idea of natural selection still arouses fierce and irrational opposition. But natural selection posed a number of problems for biology as well. What is the evidence that it operates in nature? Can it really explain adaptations, including complex ones? Darwin relied largely on analogy to make his case. The well-known success of breeders in transforming animals and plants into organisms suitable for food, pets, and decoration. But at the time he had little direct evidence for selection acting in natural populations. And because, as he proposed, selection was extremely slow, altering populations over thousands or millions of years, it would be hard to observe it acting during a single human lifetime. Fortunately, thanks to the labors of field and laboratory biologists, we now have this evidence, lots of it. Natural selection, we find, is everywhere, scrutinizing individuals, culling the unfit and promoting the genes of the fitter. It can create intricate adaptations, sometimes in surprisingly little time. Natural selection is the most misunderstood part of Darwinism. To see how it works, let's look at a simple adaptation. Coat color in wild mice. Normal-colored or old-field mice, Peromyscus polynotus, have brown coats and burrow in dark soils. But on the pale sand dunes of Florida's Gulf Coast lives a light-colored race of the same species called beach mice. These are nearly all white with only a faint brown stripe down the back. This pale color is an adaptation to camouflage the mice from predators like hawks, owls, and herons that hunt among the white dunes. How do we know this is an adaptation? A simple, albeit slightly gruesome experiment by Donald Kaufman at Kansas State University showed that mice survive better when their fur matches the color of the soil in which they live. Kaufman built large outdoor enclosures, some with light soil and others with dark soil. In each cage he put equal numbers of mice with dark and light coat colors. He then released a very hungry owl into each cage, returning later to see which mice survived. As expected, mice whose coats contrasted most conspicuously with the soil were picked off more readily, showing that camouflaged mice really do survive better. This experiment also explains a general correlation that we see in nature. Darker soils harbor darker mice. Since white color is unique among beach mice, they presumably evolved from brown mainland mice, possibly as recently as 6,000 years ago, when the barrier islands and their white dunes were first isolated from the mainland. This is where selection comes in. Old field mice vary in coat color, and among those that invaded the light beach sand, individuals with a lighter coat would have had a higher chance of surviving than darker mice, who are easily spotted by predators. We also know that there is a genetic difference between light and dark mice. Beach mice carry the light forms of several pigmentation genes that together give them their light-colored coats. Darker old-field mice have the dark alternative form of the same genes. Over time, due to the differential predation, lighter mice would have left more copies of their light genes. They have a higher chance of surviving to reproduce. And as this process continued for generation after generation, the population of beach mice would have evolved from dark to light. What happened here? Natural selection, acting on coat color, has simply changed the genetic composition of a population, increasing the proportion of genetic variants, the light-colored genes, that enhance survival and reproduction. And while I said that natural selection acts, this is not really accurate. Selection is not a mechanism imposed on a population from outside. Rather, it is a process, a description of how genes that produce better adaptations become more frequent over time. When biologists say that selection is acting on a trait, they're merely using shorthand to say that the trait is undergoing the process. In the same sense, species don't try to adapt to their environment. There is no will involved, no conscious striving. Adaptation to the environment is inevitable if a species has the right kind of genetic variation. 3 things are involved in creating an adaptation by natural selection. First, the starting population has to be variable. Mice within a population have to show some difference in their coat colors. Otherwise, this trait cannot evolve. In the case of mice, we know this is true because mice within mainland populations show some variability in coat color. Second, some proportion of that variation has to come from changes in the forms of genes. That is, the variation has to have some genetic basis called heritability. If there were no genetic difference between light and dark mice, the light ones would still survive better on the dunes, but the coat color difference would not be passed on to the next generation, and there would be no evolutionary change. We know that the genetic requirement is also satisfied in these mice. In fact, we know exactly which 2 genes have the largest effect on the dark-light color difference. One of them is called agouti, the same gene whose mutations produce black color in domestic cats. The other is called MC1R, and one of its mutant forms in humans, especially common in Irish populations, produces freckles and red hair. Where does this genetic variation come from? Mutations, accidental changes in the sequence of DNA that usually occur as errors when the molecule is copied during cell division. Genetic variation generated by mutation is widespread. Mutant forms of genes, for example, explain variation in human eye color, blood type, and much of our and other species' variation in height, weight, biochemistry, and innumerable other traits. On the basis of many laboratory experiments, scientists have concluded that mutations occur randomly. The term random here has a specific meaning that is often misunderstood, even by biologists. What this means is that mutations occur regardless of whether they would be useful to the individual. Mutations are simply errors in DNA replication. Most of them are harmful or neutral, but a few can turn out to be useful. The useful ones are the raw material for evolution. But there is no known biological way to jack up the probability that a mutation will meet the current adaptive needs of the organism. Although it's better for mice living on sand dunes to have lighter coats, their chance of getting such a useful mutation is no higher than for mice living on dark soil. Rather than calling mutations random, then, it is more accurate to call them indifferent. The chance of a mutation arising is indifferent to whether it would be helpful or hurtful to the individual. The third and last aspect of natural selection is that the genetic variation must affect an individual's probability of leaving offspring. In the case of mice, Kaufman's predation experiments showed that the most camouflaged mice would leave more copies of their genes. The white color of beach mice, then, meets all the criteria for having evolved as an adaptive trait. Evolution by selection, then, is a combination of randomness and lawfulness. There is first a random or indifferent process, the occurrence of mutations that generate an array of genetic variants, both good and bad, in the mouse example, a variety of new coat colors, and then a lawful process, natural selection, that orders this variation, keeping the good and winnowing the bad. On the dunes, light-colored genes increase at the expense of dark-color ones. This brings up what is surely the most widespread misunderstanding about Darwinism, the idea that, in evolution, everything happens by chance, also stated as everything happens by accident. This common claim is flatly wrong. No evolutionist, and certainly not Darwin, ever argued that natural selection is based on chance. Quite the opposite. Could a completely random process alone make the hammering woodpecker, the tricky bee orchid, or the camouflaged katydids and beach mice? Of course not. If suddenly evolution was forced to depend on random mutations alone, species would quickly degenerate and go extinct. Chance alone cannot explain the marvelous fit between individuals and their environment. And it doesn't. True, the raw materials for evolution, the variations between individuals, are indeed produced by chance mutations. These mutations occur willy-nilly, regardless of whether they are good or bad for the individual. But it is the filtering of that variation by natural selection that produces adaptations, and natural selection is manifestly not random. It is a powerful molding force, accumulating genes that have a greater chance of being passed on than others, and in so doing, making individuals ever better able to cope with their environment. It is, then, the unique combination of mutation and selection, chance and lawfulness, that tells us how organisms become adapted. Richard Dawkins provided the most concise definition of natural selection. It is the non-random survival of random variants. The theory of natural selection has a big job, the biggest in biology. Its task is to explain how every adaptation evolved, step by step, from traits that preceded it. This includes not just body form and color, but the molecular features that underlie everything. Selection must explain the evolution of complex physiological traits, the clotting of blood, the metabolic systems that transform food into energy, the marvelous immune system that can recognize and destroy thousands of foreign proteins. And what about the details of genetics itself? Why do pairs of chromosomes separate when eggs and sperm are formed? Why do we have sex at all instead of budding off clones, as some species do? Selection has to explain behaviors, both cooperative and antagonistic. Why do lions hunt cooperatively in a pack, and yet when intruding males displace resident males from a social group? Why do the intruders kill all the unweaned cubs? And selection has to mold these features in a particular way. First, it has to create them, most often gradually, step by step from precursors. As we have seen, each newly evolved trait begins as a modification of an earlier feature. The legs of tetrapods, for example, are simply modified fins. And each step of the process, each elaboration of an adaptation, must confer a reproductive benefit on individuals possessing it. If this doesn't happen, selection won't work. What were the advantages of each step in the transition from a swimming fin to a walking leg, or from an unfeathered dinosaur to one having both feathers and wings? There is no going downhill in the evolution of an adaptation, for selection by its very nature cannot create a step that doesn't benefit its possessor. In the world of adaptation, we never see the sign that's the bane of freeway drivers: a temporary inconvenience, a permanent improvement. If an adaptive trait evolved by natural selection instead of having been created, we can make some predictions. First, in principle, we should be able to imagine a plausible step-by-step scenario for the evolution of that trait, with each step raising the fitness, that is, the average number of offspring, of its possessor. For some traits, this is easy, like the gradual alteration of the skeleton that turned land animals into whales. For others, it is harder, especially for the biochemical pathways that leave no trace in the fossil record. We may never have enough information to reconstruct the evolution of many traits, or even in extinct species, to understand precisely how those traits functioned. What were the bony plates on the back of the Stegosaurus really for? It is telling, however, that biologists haven't found a single adaptation whose evolution absolutely requires an intermediate step that reduces the fitness of individuals. Here's another requirement. An adaptation must evolve by increasing the reproductive output of its possessor. For it is reproduction, not survival, that determines which genes make it to the next generation and cause evolution. Of course, passing on a gene requires that you first survive to the age at which you can have offspring. On the other hand, a gene that knocks you out after reproductive age incurs no evolutionary disadvantage. It will remain in the gene pool. It follows that a gene will actually be favored if it helps you reproduce in your youth, but kills you in your old age. The accumulation of such genes by natural selection, in fact, is widely thought to explain why we deteriorate in so many ways, senesce, as we reach old age. The very genes that help you sow your wild oats when young may give you wrinkles and an enlarged prostate gland later in life. Given how natural selection works, it shouldn't produce adaptations that help an individual survive without also promoting reproduction. One example would be a gene that helps human females survive after menopause. Nor do we expect to see adaptations in one species that benefit only members of another species. We can test this last prediction by looking at traits of one species that are useful to members of a second species. If those features arose by selection, we'd predict that they'll also be useful for the first species. Take tropical acacia trees, which have swollen, hollow thorns that act as homes for colonies of fierce, stinging ants. The trees also secrete nectar and produce protein-rich bodies on their leaves that provide the ants with food. It looks as if the tree is housing and feeding the ants at its own expense. Does this violate our prediction? Not at all. In fact, harboring ants gives the tree huge benefits. First, herbivorous insects and mammals that stop by for a leafy treat are repelled by a furious ant horde, as I discovered to my chagrin when brushing up against an acacia in Costa Rica. The ants also cut down seedlings around the base of the tree, seedlings which, when larger, could compete with the tree for nutrients and light. It is easy to see how acacias that were able to enlist ants to defend them from both predators and competitors would produce more seeds than acacias lacking this ability. In every case, when one species does something to help another, it always helps itself. This is a direct prediction of evolution, and one that does not follow from the notion of special creation or intelligent design. And adaptations always increase the fitness of the individual, not necessarily of the group or the species. The idea that natural selection acts for the good of the species, though common, is misguided. In fact, evolution can produce features that, while helping an individual, harm the species as a whole. When a group of male lions displaces the resident males of a pride, this is often followed by a gruesome slaughter of the unweaned cubs. This behavior is bad for the species since it reduces the total number of lions, increasing their likelihood of extinction. But it's good for the invading lions, as they can quickly fertilize the females, who come back into estrus when they're not nursing, and replace the slaughtered cubs with their own offspring. It is easy, though unsettling, to see how a gene causing infanticide would spread at the expense of nicer genes, which would have the invading males simply babysit the unrelated cubs. As evolution predicts, we never see adaptations that benefit the species at the expense of the individual, something that we might have expected if organisms were designed by a beneficent creator. Evolution without selection. Let's take a brief digression here, because it's important to appreciate that natural selection isn't the only process of evolutionary change. Most biologists define evolution as a change in the proportion of alleles, different forms of a gene, in a population. As the frequency of light color forms of the agouti gene increases in a mouse population, for example, the population and its coat color evolve. But such change can happen in other ways too. Every individual has 2 copies of each gene, which can be identical or different. Every time sexual reproduction occurs, one member of each pair of genes from a parent makes it into the offspring, along with one from the other parent. It's a toss-up which one of each parent's pair gets to the next generation. If you have an AB blood type, for example, one A allele and one B allele, and produce only one child, there's only a 50% chance it will get your A allele and a 50% chance it gets the B allele. In a one-child family, it's a certainty that one of your alleles will be lost. The upshot is that every generation, the genes of parents take part in a lottery whose prize is representation in the next generation. Because the number of offspring is finite, the frequencies of the genes present in the offspring won't be present in exactly the same frequencies as in their parents. This sampling of genes is precisely like tossing a coin. Although there is a 50% chance of getting heads on any given toss, if you make only a few tosses, there is a substantial chance that you'll deviate from this expectation. In 4 tosses, for example, you have a 12% chance of getting either all heads or all tails. And so, especially in small populations, the proportion of different alleles can change over time entirely by chance. And new mutations may enter the fray and themselves rise or fall in frequency due to this random sampling. Eventually, the resulting random walk can even cause genes to become fixed in the population, that is, rise to 100% frequency, or, alternatively, get completely lost. Such random change in the frequency of genes over time is called genetic drift. It is a legitimate type of evolution, since it involves changes in the frequencies of alleles over time, but it doesn't arise from natural selection. One example of evolution by drift may be the unusual frequencies of blood types, as in the ABO system, in the Old Order Amish and Dunker religious communities in America. These are small, isolated religious groups whose members intermarry, just the right circumstances for rapid evolution by genetic drift. Accidents of sampling can also happen when a population is founded by just a few immigrants, as occurs when individuals colonize an island or a new area. The almost complete absence of genes producing the B blood type in Native American populations, for example, may reflect the loss of this gene in a small population of humans that colonized North America from Asia around 12,000 years ago. Both drift and natural selection produce the genetic change that we recognize as evolution. But there's an important difference. Drift is a random process, while selection is the antithesis of randomness. Genetic drift can change the frequencies of alleles regardless of how useful they are to their carrier. Selection, on the other hand, always gets rid of harmful alleles and raises the frequencies of beneficial ones. As a purely random process, genetic drift can't cause the evolution of adaptations. It could never build a wing or an eye. That takes non-random natural selection. What drift can do is cause the evolution of features that are neither useful nor harmful to the organism. Ever prescient, Darwin himself broached this idea in The Origin. This preservation of favorable variations and the rejection of injurious variations I call natural selection. Variations neither useful nor injurious would not be affected by natural selection, and would be left as a fluctuating element, as perhaps we see in the species called polymorphic. In fact, genetic drift is not only powerless to create adaptations, but can actually overpower natural selection. Especially in small populations, the sampling effect can be so large that it raises the frequency of harmful genes, even though selection is working in the opposite direction. This is almost certainly why we see a high incidence of genetically based diseases in isolated human communities, including Gaucher's disease in northern Swedes, Tay-Sachs in the Cajuns of Louisiana, and retinitis pigmentosa in the inhabitants of the island of Tristan da Cunha. Because certain variations in DNA or protein sequence may be, as Darwin put it, neither useful nor injurious, or neutral as we now call them, such variants are especially liable to evolve by drift. For example, some mutations in a gene don't affect the sequence of the protein that it produces, and so don't change the fitness of its carrier. The same goes for mutations in non-functioning pseudogenes, old wrecks of genes still kicking around in the genome. Any mutations in these genes have no effect on the organism and therefore can evolve only by genetic drift. Many aspects of molecular evolution, then, such as certain changes in DNA sequence, may reflect drift rather than selection. It's also possible that many externally visible features of organisms could evolve via drift, especially if they don't affect reproduction. The diverse shapes of leaves of different tree species, like the differences between oak and maple leaves, were once suggested to be neutral traits that evolved by genetic drift. But it's hard to prove that a trait has absolutely no selective advantage. Even a tiny advantage, so small as to be unmeasurable or unobservable by biologists in real time, can lead to important evolutionary change over eons. The relative importance of genetic drift versus selection in evolution remains a topic of hot debate among biologists. Every time we see an obvious adaptation, like the camel's hump, we clearly see evidence for selection. But features whose evolution we don't understand may reflect only our ignorance rather than genetic drift. Nevertheless, we know that genetic drift must occur, because in any population of finite size, there are always sampling effects during reproduction. And drift has probably played a substantial role in the evolution of small populations, although we can't point to more than a few examples. Animal and plant breeding. The theory of natural selection predicts what types of adaptations we'd expect to find and, more important, not find in nature. And these predictions have been fulfilled. But many people would like more. They'd like to see natural selection in action and witness evolutionary change in their lifetime. It's not hard to accept the idea that natural selection could cause, say, the evolution of whales from land animals over millions of years. But somehow the idea of selection becomes more compelling when we see the process act before our eyes. This demand to see selection and evolution in real time, while understandable, is curious. After all, we easily accept that the Grand Canyon resulted from millions of years of slow, imperceptible carving by the Colorado River, even though we can't see the canyon getting deeper over our lifetime. But for some people, this ability to extrapolate time for geological forces doesn't apply to evolution. How, then, can we determine whether selection has been an important cause of evolution? Obviously, we can't replay the evolution of whales to see the reproductive advantage of each small step that took them back to the water. But if we can see selection causing small changes over just a few generations, then it becomes easier to accept that, over millions of years, similar types of selection could cause the big adaptive changes documented in fossils. Evidence for selection comes from many areas, The most obvious is artificial selection, animal and plant breeding, which, as Darwin realized, is a good parallel to natural selection. We know that breeders have worked wonders in transforming wild plants and animals into completely different forms that are good to eat or that satisfy our aesthetic needs. And we know that this has been done by selecting variation present in their wild ancestors. We also know that breeding has wrought huge changes in a remarkably short period of time, for animal and plant breeding has been practiced for only a few thousand years. Take the domestic dog, Canis lupus familiaris, a single species that comes in all shapes, sizes, colors, and temperaments. Every single one, purebred or mutt, descends from a single ancestral species, most likely the Eurasian gray wolf, that humans began to select about 10,000 years ago. The American Kennel Club recognizes 150 different breeds, and you've seen many of them. The tiny nervous Chihuahua, perhaps bred as a food animal by the Toltec of Mexico. The robust St. Bernard, thick of fur and able to carry kegs of brandy to snow-stranded travelers. The Greyhound, bred for racing with long legs and a streamlined shape. The elongated, short-legged Dachshund, ideal for catching badgers in their holes. Retrievers, bred to fetch game from the water. And the fluffy Pomeranian, bred as a comforting lapdog. Breeders have virtually sculpted these dogs to their liking, changing the shade and thickness of their coats, the length and pointiness of their ears, the size and shape of their skeletons, the quirks of their behaviors and temperaments, and nearly everything else. Think of the diversity you'd see if all these dogs were lined up together. If somehow the recognized breeds existed only as fossils, paleontologists would consider them not one species but many. Certainly more than the 36 species of wild dogs that live in nature today. In fact, the variation among domestic dogs far exceeds that among wild dog species. Take just one trait: weight. Domestic dogs range from the 2-pound Chihuahua to the 180-pound English Mastiff, while the weight of wild dog species varies from 2 pounds to only about 60 pounds. And there is certainly no wild dog having the shape of a dachshund or the face of a pug. The success of dog breeding validates 2 of the 3 requirements for evolution by selection. First, there was ample variation in color, size, shape, and behavior in the ancestral lineage of dogs to make possible the creation of all breeds. Second, some of that variation was produced by genetic mutations that could be inherited. For if it were not, breeders could make no progress. What is most astonishing about dog breeding is how fast it got results. All those breeds have been selected in less than 10,000 years, only 0.1% of the time that it took wild dog species to diversify from their common ancestor in nature. If artificial selection can produce such canine diversity so quickly, it becomes easier to accept that the lesser diversity of wild dogs arose by natural selection, acting over a period 1,000 times longer. There's really only one difference between artificial and natural selection. In artificial selection, it is the breeder rather than nature who sorts out which variants are good and bad. In other words, the criterion of reproductive success is human desire rather than adaptation to a natural environment. Sometimes these criteria coincide. Look, for example, at the greyhound, which was selected for speed and wound up shaped very much like a cheetah. This is an example of convergent evolution. Similar selective pressures give similar outcomes. The dog can stand for the success of other breeding programs. As Darwin noted in The Origin, breeders habitually speak of an animal's organization as something quite plastic, which they can model almost as they please. Cows, sheep, pigs, flowers, vegetables, and so on, all came from humans choosing variants present in wild ancestors, or variants that arose by mutation during domestication. Through selection, The svelte wild turkey has become our docile, meaty, and virtually tasteless Thanksgiving monster, with breasts so large that male domestic turkeys can no longer mount females, who must instead be artificially inseminated. Darwin himself bred pigeons and described the huge variety of behaviors and appearance of different breeds, all selected from the ancestral rock dove. You wouldn't recognize the ancestor of our ear of corn, which was an inconspicuous grass. The ancestral tomato weighed only a few grams, but has now been bred into a 2-pound behemoth, also tasteless, with a long shelf life. The wild cabbage has given rise to 5 different vegetables: broccoli, domestic cabbage, kohlrabi, Brussels sprouts, and cauliflower, each selected to modify a different part of the plant. Broccoli, for example, is simply a tight, enlarged cluster of flowers. And the domestication of all wild crop plants occurred within the last 12,000 years. It's no surprise, then, that Darwin began the origin not with a discussion of natural selection or evolution in the wild, but with a chapter called Variation Under Domestication, on animal and plant breeding. He knew that if people could accept artificial selection, and they had to because its success was so obvious, then making the leap to natural selection was not so hard. As he argued, under domestication, it may be truly said that the whole organization becomes in some degree plastic. Can it then be thought improbable, seeing that variations useful to man have undoubtedly occurred, that other variations useful in some way to each being in the great and complex battle of life should sometimes occur in the course of thousands of generations? Since domestication of wild species took place only in the relatively short period since humans became civilized, Darwin knew that it wouldn't be much of a stretch to accept that natural selection could create much greater diversity over a much longer time. Evolution in the test tube. We can go a step further. Instead of breeders picking out favored variants, we can let this happen naturally in the laboratory by exposing a captive population to new environmental challenges. This is easiest to do in microbes like bacteria, which can divide as often as once every 20 minutes, allowing us to observe evolutionary change over thousands of generations in real time. And this is genuine evolutionary change, demonstrating all 3 requirements of evolution via selection: variation, heritability, and the differential survival and reproduction of variants. Although the environmental challenge is created by humans, these sorts of experiments are more natural than artificial selection because humans don't choose which individuals get to reproduce. Let's start with simple adaptations. Microbes can adapt to virtually anything that scientists throw at them in the lab: high or low temperature, antibiotics, toxins, starvation, new nutrients, and their natural enemies, viruses. Probably the longest-running study of this type has been carried out by Richard Lenski at Michigan State University. In 1988, Lenski put genetically identical strains of the common gut bacterium E. coli under conditions in which their food, the sugar glucose, was depleted each day and then renewed the next. This experiment was thus a test of the microbe's ability to adapt to a feast and famine environment. Over the next 18 years, 40,000 bacterial generations, the bacteria continued to accumulate new mutations, adapting them to this new environment. Under the varying food conditions, they now grow 70% faster than the original unselected strain. The bacteria continue to evolve, and Lenski and his colleagues have identified at least 9 genes whose mutations result in adaptation. But laboratory adaptations can also be more complex, involving the evolution of whole new biochemical systems. Perhaps the ultimate challenge is simply to take away a gene that a microbe needs to survive in a particular environment and see how it responds. Can it evolve a way around this problem? The answer is usually yes. In a dramatic experiment, Barry Hall and his colleagues at the University of Rochester began a study by deleting a gene from E. coli. This gene produces an enzyme that allows the bacteria to break down the sugar lactose into subunits that can be used as food. The geneless bacteria were then put in an environment containing lactose as the only food source. Initially, of course, they lacked the enzyme and couldn't grow. But after only a short time, the function of the missing gene was taken over by another enzyme that, while previously unable to break down lactose, could now do so weakly because of a new mutation. Eventually, yet another adaptive mutation occurred, one that increased the amount of the new enzyme so that even more lactose could be used. Finally, a third mutation at a different gene allowed the bacteria to take up lactose from the environment more easily. Altogether, this experiment showed the evolution of a complex biochemical pathway that enabled bacteria to grow on a previously unusable food. Beyond demonstrating evolution, this experiment has two important lessons. First, natural selection can promote the evolution of complex, interconnected biochemical systems in which all the parts are codependent, despite the claims of creationists that this is impossible. Second, as we've seen repeatedly, selection does not create new traits out of thin air. It produces new adaptations by modifying pre-existing features. We can even see the origin of new, ecologically diverse bacterial species, all within a single laboratory flask. Paul Rainey and his colleagues at Oxford University placed a strain of the bacteria, Pseudomonas fluorescens, in a small vessel containing nutrient broth and simply watched it. It's surprising but true that such a vessel actually contains diverse environments. Oxygen concentration, for example, is highest on the top and lowest on the bottom. Within 10 days, no more than a few hundred generations, the ancestral, free-floating, smooth bacterium had evolved into two additional forms occupying different parts of the beaker. One, called wrinkly spreader, formed a mat on top of the broth. The other, called fuzzy spreader, formed a carpet on the bottom. The smooth ancestral type persisted in the liquid environment in the middle. Each of the two new forms was genetically different from the ancestor, having evolved through mutation and natural selection to reproduce best in their respective environments. Here, then, is not only evolution but speciation occurring in the lab. The ancestral form produced and coexisted with two ecologically different descendants. And in bacteria, such forms are considered distinct species. Over a very short time, natural selection on Pseudomonas yielded a small-scale adaptive radiation, the equivalent of how animals or plants form species when they encounter new environments on an oceanic island. Resistance to drugs and poisons. When antibiotics were first introduced in the 1940s, everyone thought that they would finally solve the problem of infectious disease caused by bacteria. The drugs worked so well that nearly everyone with tuberculosis, strep throat, or pneumonia could be cured with a couple of simple injections or a vial of pills. But we forgot about natural selection. Given their huge population sizes and short generation times, features that make bacteria ideal for studies of evolution in the lab, the chance of a mutation producing antibiotic resistance is high. And those bacteria that are resistant to a drug will be those that survive, leaving behind genetically identical offspring that are also drug-resistant. Eventually, the effectiveness of the drug wanes, and once again we have a medical problem. This has become a severe crisis for some diseases. There are now strains of tuberculosis bacteria, for example, that have evolved resistance to every drug doctors have used against them. After a long period of cures and medical optimism, TB is once again becoming a fatal disease. This is natural selection, pure and simple. Everyone knows about drug resistance, But it's not often realized that this is about the best example we have of selection in action. Had this phenomenon existed in Darwin's time, he would certainly have made it a centerpiece of The Origin. It is a widespread belief that drug resistance occurs because somehow the patients themselves change in a way that makes the drug less effective. But this is wrong. Resistance comes from evolution of the microbe, not habituation of patients to the drugs. Another prime example of selection is resistance to penicillin. When it was introduced in the early 1940s, penicillin was a miracle drug, especially effective at curing infections caused by the bacteria Staphylococcus aureus, staph. In 1941, the drug could wipe out every strain of staph in the world. Now, 70 years later, more than 95% of staph strains are resistant to penicillin. What happened was that mutations occurred in individual bacteria that gave them the ability to destroy the drug. And of course, these mutations spread worldwide. In response, the drug industry came up with a new antibiotic, methicillin. But even that is now becoming useless due to newer mutations. In both cases, scientists have identified the precise changes in the bacterial DNA that conferred drug resistance. Viruses, the smallest form of evolvable life, have also evolved resistance to antiviral drugs, most notably AZT, azithromycin, designed to prevent the HIV virus from replicating in an infected body. Evolution even occurs within the body of a single patient, since the virus mutates at a furious pace, eventually producing resistance and rendering AZT ineffective. Now we keep AIDS at bay with a daily 3-drug cocktail, and if history is any guide, this too will eventually stop working. The evolution of resistance creates an arms race between humans and microorganisms, in which the winners are not just bacteria, but also the pharmaceutical industry, which constantly devises new drugs to overcome the waning effectiveness of old ones. But fortunately, there are some spectacular cases of microorganisms that haven't succeeded in evolving resistance. We must remember that the theory of evolution doesn't predict that everything will evolve. If the right mutations can't or don't arise, evolution won't happen. One form of streptococcus, for example, causes strep throat, a common infection in children. These bacteria have failed to evolve even the slightest resistance to penicillin, which remains the treatment of choice. And unlike the influenza virus, polio and measles viruses have not evolved resistance to the vaccines that have now been used for over 50 years. Still other species have adapted via selection to human-caused changes in their environment. Insects have become resistant to DDT and other pesticides. Plants have adapted to herbicides, and fungi, worms, and algae have evolved resistance to heavy metals that have polluted their environment. There almost always seem to be a few individuals with lucky mutations that allow them to survive and reproduce, quickly evolving a sensitive population into a resistant one. We can then make a reasonable inference. When a population encounters a stress that doesn't come from humans, such as a change in salinity, temperature, or rainfall, natural selection will often produce an adaptive response. Selection in the wild. The responses we've seen to human-imposed stress and chemicals constitute natural selection in any meaningful sense. Although the selective agents are devised by humans, the response is purely natural and, as we've seen, can be quite complex. But perhaps it would be even more convincing to see the whole process in action in nature without human intervention. That is, we want to see a natural population meet a natural challenge. We want to know what that challenge is, and we want to see the population evolve to meet it before our eyes. We can't expect this circumstance to be common. For one thing, natural selection in the wild is often incredibly slow. The evolution of feathers, for example, probably took hundreds of thousands of years. Even if feathers were evolving today, it would simply be impossible to watch this happening in real time, much less to measure whatever type of selection was acting to make feathers larger. If we are to see natural selection at all, it must be strong selection causing rapid change, and we'd best look at animals or plants having short generation times so that the evolutionary change can be seen over several generations. And we have to do better than bacteria. People want to see selection in so-called higher plants and animals. Further, we shouldn't expect to see more than small changes in one or a few features of a species, what is known as microevolutionary change. Given the gradual pace of evolution, it's unreasonable to expect to see selection transforming one type of plant or animal into another, so-called macroevolution, within a human lifetime. Though macroevolution is occurring today, we simply won't be around long enough to see it. Remember that the issue is not whether macroevolutionary change happens. We already know from the fossil record that it does, but whether it was caused by natural selection, and whether natural selection can build complex features and organisms. Another factor making it hard to see real-time selection is that a very common type of natural selection doesn't cause species to change. Every species is pretty well adapted, which means that selection has already brought it into sync with its environment. Episodes of change that occur when a species meets a new environmental challenge are probably rare compared to periods when there's nothing new to adapt to. But that doesn't mean that selection is not occurring. If a species of birds, for example, has evolved the optimum body size for its environment, and that environment doesn't change, selection will act only to cull birds that are larger or smaller than the optimum. But this kind of selection, called stabilizing selection, won't change the average body size. If you look at the population from one generation to the next, nothing much will have changed, although genes for both large and small sizes will have been eliminated. We can see this, for example, for birth weight in human babies. Hospital statistics consistently show that babies having average birth weights, around 7.5 pounds in the United States and Europe, survive better than either lighter babies, born prematurely or from malnourished mothers, or heavier babies, who have difficulties being born. If we want to see selection in action, then we should look in species that have short generation times and are adapting to a new environment. This is most likely to happen when a species either invade a new habitat or experience severe environmental change. And indeed, that is where the examples lie. The most famous of these, which I won't belabor as it's been described in detail elsewhere, see, for example, Jonathan Weiner's superb book, The Beak of the Finch, A Story of Evolution in Our Time, is the adaptation of a bird to an anomalous change in climate. The medium ground finch of the Galapagos Islands has been studied for several decades by Peter and Rosemary Grant of Princeton University and their colleagues. In 1977, a severe drought in the Galapagos drastically reduced the supply of seeds on the island of Daphne Major. This finch, which normally prefers small, soft seeds, was forced to turn to larger and harder ones. Experiments showed that hard seeds are easily cracked only by larger birds, which have bigger and stouter beaks. The upshot was that only big-beaked individuals got adequate food, while those with smaller beaks starved to death or were too malnourished to reproduce. The large-beaked survivors left more offspring, and by the next generation, natural selection had increased the average beak size by 10%. Body size increased as well. This is a staggering rate of evolutionary change, far larger than anything we see in the fossil record. In comparison, brain size in the human lineage increased on average about 0.001% per generation. Everything we require of evolution by natural selection was amply documented by the Grants in other studies. Individuals in the original population varied in beak depth. A large proportion of that variation was genetic, and individuals with different beaks left different numbers of offspring in the predicted direction. Given the importance of food to survival, The ability to gather, eat, and digest it efficiently is a strong selective force. Many insects are host-specific. They feed and lay their eggs on only one or a few species of plants. In such cases, the insect needs adaptations for using the plants, including the right feeding apparatus to tap the plant's nutrients, a metabolism that detoxifies any plant poisons, and a reproductive cycle that produces young when there is available food, the plant's fruiting period. Since there are many closely related pairs of insects that use different host plants, there must have been many switches from one plant to another over evolutionary time. These switches, equivalent to colonizing a very different habitat, must have been accompanied by strong selection. We have in fact seen this happen over the last few decades in the soapberry bug, Chidera hematoloma, of the New World. Chidera lives on two native plants in different parts of the United States, the soapberry bush in the south-central U.S. and the perennial balloon vine in southern Florida. With its long, needle-like beak, the bug penetrates the fruits of these plants and consumes the seeds within, liquefying their contents and sucking them up. But within the last 50 years, the bug has colonized 3 other plants introduced into its range. The fruits of these plants are very different in size from those of its native host. 2 are much larger and 1 much smaller. Scott Carroll and his colleagues predicted that this host switch would cause natural selection for changes in beak size. Bugs colonizing the larger fruited species should evolve larger beaks to penetrate the fruits and reach the seeds, while bugs colonizing the smaller fruited species would evolve in the opposite direction. This is exactly what happened. With beak length changing by up to 25% in a few decades. This may not seem like much, but it is enormous by evolutionary standards, particularly over the short span of 100 generations. To put it in perspective, if this rate of beak evolution was sustained over only 10,000 generations, 5,000 years, the beaks would increase in size by a factor of roughly 5 billion, becoming about 1,800 miles long and able to skewer a fruit the size of the moon. This ludicrous and unrealistic figure is, of course, meant only to show the cumulative power of seemingly small changes. Here's another prediction. Under prolonged drought, natural selection will lead to the evolution of plants that flower earlier than their ancestors. This is because during a drought, soils dry out quickly after the rains. If you're a plant that doesn't flower and produce seeds quickly in a drought, you leave no descendants. Under normal weather conditions, on the other hand, it pays to delay flowering so that you can grow larger and produce even more seeds. This prediction was tested in a natural experiment involving the wild mustard plant, Brassica rapa, introduced to California about 300 years ago. Beginning in 2000, Southern California suffered a severe 5-year drought. Arthur Weiss and his colleagues at the University of California measured the flowering time of mustards at the beginning and end of this period. Sure enough, natural selection had changed flowering time in precisely the predicted way. After the drought, plants began to flower a week earlier than their ancestors did. There are many more examples, but they all demonstrate the same thing. We can directly witness natural selection leading to better adaptation. Natural Selection in the Wild, a book by the biologist John Endler, documents over 150 cases of observed evolution, and in roughly a third of these we have a good idea about how natural selection was acting. We see fruit flies adapting to extreme temperature, honeybees adapting to competitors, and guppies becoming less colorful to escape the notice of predators. How many more examples do we need? Can selection build complexity? But even if we agree that natural selection does work in nature, how much work can it really do? Sure, selection can change the beaks of birds or the flowering period of plants, but can it build complexity? What about intricate traits like the tetrapod limb, or exquisite biochemical adaptations like blood clotting, which entails a precise sequence of steps involving many proteins, or perhaps the most complicated apparatus that ever evolved, the human brain. We're at somewhat of a handicap here because, as we know, complex features take a long time to evolve, and most of them did so in the distant past when we weren't around to see how it happened. So how can we be sure that selection was involved? How do we know that creationists are wrong when they say that selection can make small changes in organisms, but is powerless to make big ones? But first we must ask, what's the alternative theory? We know of no other natural process that can build a complex adaptation. The most commonly suggested alternative takes us into the realm of the supernatural. This, of course, is creationism, known in its latest incarnation as intelligent design. Advocates of ID suggest that a supernatural designer has intervened at various times during the history of life, either instantly calling into being the complex adaptations that natural selection supposedly can't make, or producing miracle mutations that can't occur by chance. Some ideas go further. They are the extreme young Earth creationists who believe that Earth is about 6,000 years old and that life has no evolutionary history at all. In the main, ID is unscientific, for it consists largely of untestable claims. How, for example, can we determine whether mutations were mere accidents in DNA replication or were willed into being by a creator? But we can still ask if there are adaptations that could not have been built by selection, and therefore require us to think of another mechanism. Advocates of ID have suggested several such adaptations, such as the bacterial flagellum, a small hair-like apparatus with a complex molecular motor used by some bacteria to propel themselves, and the mechanism of blood clotting. These are indeed complex features. The flagellum, for instance, is composed of dozens of separate proteins, all of which must work in concert for the hair-like propeller to move. Ideers argue that such traits, involving many parts that must cooperate for that trait to function at all, defy Darwinian explanation. Therefore, by default, they must have been designed by a supernatural agent. This is commonly called the God of the Gaps argument, and it is an argument from ignorance. What it really says is that if we don't understand everything about how natural selection built a trait, that lack of understanding itself is evidence for supernatural creation. You can probably see why this argument doesn't hold water. We'll never be able to reconstruct how selection created everything. Evolution happened before we were on the scene, and some things will always be unknown. But evolutionary biology is like every science. It has mysteries, and many of them get solved one after the other. We now know, for instance, where birds came from. They weren't created out of thin air, as creationists used to maintain, but evolved gradually from dinosaurs. And each time a mystery is solved, ID is forced to retreat. Since ID itself makes no testable scientific claims, but offers only half-baked criticisms of Darwinism, its credibility slowly melts away with each advance in our understanding. Further, ID's own explanation for complex features, the whim of a supernatural designer, can explain any conceivable observation about nature. It may even have been the Creator's whim to make life look as though it evolved. Apparently, many creationists believe this, though few admit it. But if you can't think of an observation that could disprove a theory, that theory simply isn't scientific. How, though, can we refute the ID claim that some traits simply defy any origin by natural selection? In such cases, the onus is not on evolutionary biologists to sketch out a precise step-by-step scenario documenting exactly how a complex character evolved. That would require knowing everything about what happened when we were not around, an impossibility for most traits and for nearly all biochemical pathways. As the biochemists Ford Doolittle and Olga Shaksabayeva argued when addressing the ID claim that flagella could not have evolved, evolutionists need not take on the impossible challenge of pinning down every detail of flagellar evolution, We need only show that such a development, involving processes and constituents not unlike those we already know and can agree upon, is feasible. And by feasible, they mean that there must be evolutionary precursors of each new trait, and that evolution of that trait does not violate the Darwinian requirement that each step in building an adaptation benefits its possessor. Indeed, we know of no adaptations whose origin could not have involved natural selection. How can we be sure? For anatomical traits, we can simply trace their evolution, when possible, in the fossil record, and see in what order different changes took place. We can then determine whether the sequence of changes at least conform to a step-by-step adaptive process. And in every case, we can find at least a feasible Darwinian explanation. We've seen this for the evolution of land animals from fish, of whales from land animals, and of birds from reptiles. It didn't have to be that way. The movement of nostrils to the top of the head in ancestral whales, for example, could have preceded the evolution of fins. That could be the providential act of a creator, but couldn't have evolved by natural selection. But we always see an evolutionary order that makes Darwinian sense. Understanding the evolution of complex biochemical features and pathways is not as easy, since they leave no trace in the fossil record. Their evolution must be reconstructed in more speculative ways, trying to see how such pathways could be cobbled together from simpler biochemical precursors. And we'd like to know the steps in this cobbling to see if each new one could bring improved fitness. Although advocates of ID claim a supernatural hand behind these pathways, dogged scientific research is beginning to give plausible and testable scenarios for how they could have evolved. Take the blood clotting pathway of vertebrates. This involves a sequence of events that begins when one protein sticks to another in the vicinity of an open wound. That sets off a complicated cascade reaction 16 steps long, each involving an interaction between a different pair of proteins and culminating in the formation of the clot itself. Altogether, more than 20 proteins are involved. How could this possibly have evolved? We don't know yet for sure, but we have evidence that the system could have been built up in an adaptive way from simpler precursors. Many of the blood clotting proteins are made by related genes that arose by duplication, a form of mutation in which an ancestral gene, and later its descendants, becomes duplicated in full along a strand of DNA because of a mistake during cell division. Once they arise, such duplicated genes can then evolve along separate pathways so that they eventually perform separate functions, as they now do in blood clotting. And we know that other proteins and enzymes in the pathway had different functions in groups that evolved before vertebrates. For example, a key protein in the clotting pathway is called fibrinogen, which is dissolved in blood plasma. In the last step of blood clotting, this protein gets cut by an enzyme, and the shorter proteins, called fibrins, stick together and become insoluble, forming the final clot. Since fibrinogen occurs in all vertebrates as a blood clotting protein, it presumably evolved from a protein that had a different function in ancestral invertebrates, who were around earlier but lacked a clotting pathway. Although an intelligent designer could invent a suitable protein, evolution doesn't work that way. There must have been an ancestral protein from which fibrinogen evolved. Russell Doolittle at the University of California predicted that we would find such a protein, and sure enough, in 1990 he and his colleague Zun Zhu discovered it in the sea cucumber, an invertebrate sometimes used in Chinese cooking. Sea cucumbers branched off from the vertebrate lineage at least 500 million years ago. Yet they have a protein that, while clearly related to blood-clotting proteins of vertebrates, is not used to clot blood. This means that the common ancestor of sea cucumbers and vertebrates had a gene that was later co-opted in vertebrates for a new function, precisely as evolution predicts. Since then, both Doolittle and cell biologist Ken Miller have worked out a plausible and adaptive sequence for the evolution of the entire blood clotting cascade from parts of precursor proteins. All of these precursors are found in invertebrates, where they have other, non-clotting functions, and were evolutionarily co-opted by vertebrates into a working clotting system. And the evolution of the bacterial flagellum, though not yet fully understood, is also known to involve many proteins co-opted from other biochemical pathways. Hard problems often yield before science, and though we still don't understand how every complex biochemical system evolved, we are learning more every day. After all, biochemical evolution is a field still in its infancy. If the history of science teaches us anything, it is that what conquers our ignorance is research, not giving up and attributing our ignorance to the miraculous work of a creator. When you hear someone claim otherwise, just remember these words of Darwin. Ignorance more frequently begets confidence than does knowledge. It is those who know little, and not those who know much, who so positively assert that this or that problem will never be solved by science. It appears, then, that in principle there is no real problem with evolution building complex biochemical systems. But what about time? Has there really been enough time for natural selection to create both complex adaptations as well as the diversity of living forms? Certainly we know that there was enough time for organisms to have evolved. The fossil record alone tells us that. But was natural selection strong enough to drive such change? One approach is to compare the rates of evolution in the fossil record with those seen in laboratory experiments that used artificial selection, or with historical data on evolutionary change that occurred when species colonized new habitats in historical times. If evolution in the fossil record were much faster than in laboratory experiments or colonization events, both of which involve very strong selection, we might need to rethink whether selection could explain changes in fossils. But in fact, the results are just the opposite. Philip Gingerich at the University of Michigan showed that rates of change in animal size and shape during laboratory and colonization studies are actually much faster than rates of fossil change, from 500 times faster, selection during colonizations, to nearly 1,000,000 times faster, laboratory selection experiments. And even the fastest rates of evolution in the fossil record are nowhere near as fast as the slowest rates seen when humans practice selection in the laboratory. Further, the average rates of evolution seen in colonization studies are large enough to turn a mouse into the size of an elephant in just 10,000 years. The lesson, then, is that selection is perfectly adequate to explain changes that we see in the fossil record. One reason why people raise this question is because they don't or can't appreciate the immense spans of time that selection has had to work. After all, we evolved to deal with things that happen on the scale of our lifetime, probably around 30 years during most of our evolution. A span of 10 million years is beyond our intuitive grasp. Finally, is natural selection sufficient to explain a really complex organ, such as the eye? The camera eye of vertebrates and mollusks, like the squid and octopus, was once beloved by creationists. Noting its complex arrangement of the iris, lens, retina, cornea, and so on, all of which must work together to create an image, opponents of natural selection claimed that the eye could not have formed by gradual steps. How could half an eye be of any use? Darwin brilliantly addressed and rebutted this argument in The Origin. He surveyed existing species to see if one could find functional but less complex eyes that not only were useful, but also could be strung together in a hypothetical sequence showing how a camera eye might evolve. If this could be done, and it can, then the argument that natural selection could never produce an eye collapses, for the eyes of existing species are obviously useful. Each improvement in the eye could confer obvious benefits, for it makes an individual better able to find food, avoid predators, and navigate around its environment. A possible sequence of such changes begins with simple eye spots made of light-sensitive pigment, as seen in flatworms. The skin then folds in, forming a cup that protects the eye spot and allows it to better localize the light source. Limpets have eyes like this. In the chambered nautilus, we see a further narrowing of the cup's opening to produce an improved image, and in ragworms, the cup is capped by a transparent cover to protect the opening. In abalones, part of the fluid in the eye has coagulated to form a lens, which helps focus light. And in many species, such as mammals, nearby muscles have been co-opted to move the lens and vary its focus. The evolution of a retina, an optic nerve, and so on follows by natural selection. Each step of this hypothetical transitional series confers increased adaptation on its possessor, because it enables the eye to gather more light or form better images, both of which aid survival and reproduction. And each step of this process is feasible, because it is seen in the eyes of a different living species. At the end of the sequence we have the camera eye, whose adaptive evolution seems impossibly complex. But the complexity of the final eye can be broken down into a series of small, adaptive steps. Yet we can do even better than just stringing together eyes of existing species in an adaptive sequence. We can, starting with a simple precursor, actually model the evolution of the eye and see whether selection can turn that precursor into a more complex eye in a reasonable amount of time. Dan Erik Nilsson and Suzanne Pelger of Lund University in Sweden made such a mathematical model, starting with a patch of light-sensitive cells backed by a pigment layer, a retina. They then allowed the tissues around this structure to deform themselves randomly, limiting the amount of change to only 1% of size or thickness at each step. To mimic natural selection, the model accepted only mutations that improved the visual acuity and rejected those that degraded it. Within an amazingly short time, the model yielded a complex eye, going through stages similar to the real animal series described above. The eye folded inward to form a cup. The cup became capped with a transparent surface, and the interior of the cup gelled to form not only a lens, but a lens with dimensions that produced the best possible image. Beginning with a flatworm-like eye spot, then, the model produced something like the complex eye of vertebrates, all through a series of tiny adaptive steps, 1,829 of them to be exact. But Nilsson and Pelger also calculated how long this process would take. To do this, they made some assumptions about how much genetic variation for eye shape existed in the population that began experiencing selection, and about how strongly selection would favor each useful step in eye size. These assumptions were deliberately conservative, assuming that there were reasonable but not large amounts of genetic variation, and that natural selection was very weak. Nevertheless, the eye evolved very quickly. The entire process from rudimentary light patch to camera eye took fewer than 400,000 years. Since the earliest animals with eyes date back 550 million years ago, there was, according to this model, enough time for complex eyes to have evolved more than 1,500 times over. In reality, eyes have evolved independently in at least 40 groups of animals. As Nilsson and Pelger noted dryly in their paper, it is obvious that the eye was never a real threat to Darwin's theory of evolution. So where are we? We know that a process very like natural selection, animal and plant breeding, has taken the genetic variation present in wild species and from it created huge evolutionary transformations. We know that these transformations can be much larger and faster than real evolutionary change that took place in the past. We've seen that selection operates in the laboratory, in microorganisms that cause disease, and in the wild. We know of no adaptations that absolutely could not have been molded by natural selection, and in many cases we can plausibly infer how selection did mold them. And mathematical models show that natural selection can produce complex features easily and quickly. The obvious conclusion? we can provisionally assume that natural selection is the cause of all adaptive evolution, though not of every feature of evolution, since genetic drift can also play a role. True, breeders haven't turned a cat into a dog, and laboratory studies haven't turned a bacterium into an amoeba, although, as we've seen, new bacterial species have arisen in the lab. But it is foolish to think that these are serious objections to natural selection. Big transformations take time, huge spans of it. To really see the power of selection, we must extrapolate the small changes that selection creates in our lifetime over the millions of years that it has really had to work in nature. We can't see the Grand Canyon getting deeper either, but gazing into that great abyss, with the Colorado River carving away insensibly below, you learn the most important lesson of Darwinism. Weak forces operating over long periods of time create large and dramatic change.